introduction. So what I thought like I would be covering today, the molecular microscope. And it is not a new story. It has been started long back, almost like 3 decades ago in US. So we have the pictures of Marilyn Monroe and Madhuri Dixit in here, just have some similarity. You see the H&E flavor, the same smile, H&E to next-gen sequencing. They were in the Hollywood and Bollywood in two different eras, and but they kind of try. I mean, with all these techniques, we can still make our cancer patients happy with advanced diagnostics and specifically finding out therapeutic options for them. So there is a paradigm shift in cancer. Before that, we need to know what is oncology. So these are the people with whom I work at the three centers I work. So, the revolving role of pathologist is increasing. We are kind of evolved, but there are threats and opportunities with the contemporary healthcare in the era of precision medicine. And precision healthcare or personalized medicine, best healthcare is empowering it, and digital pathology is enabling to make it one. So I have no conflict of interest to declare. And so before I start, I would like to speak a line for a patient point of view. And the patient is willing to fight the battle, but why? He should know why he is battling for and what is the need he should be doing that. So the outlines of my talk would be I'd be defining a few entities, which all of you, I'm sure, know about it, a few things about the cell cycle, the pathways for cancer, what are the diagnostic and sine qua non of malignancy, clinical implications of everything we do. Without that, it's kind of a ex vacuo approach. What are the genetic abnormalities in cancer? How can we implement those genetic data in diagnostic laboratory, and what are the clinical values? So there are a few definitions we all need to know. What is a tumor? It's a swelling which is exclusively used to denote a neoplasm. And neoplasia is a new formation or a new growth. And oncology is the study of tumor or the neoplasm, and especially the malignant neoplasm. And malignancy is synonymous with a grab or a cancer. So way back, Dr. Rupert Allen Willis, who coined the word neoplasia first. So it is an abnormal mass of tissue, the growth of which exceeds, and it is uncoordinated with that of the normal tissue or the normal counterpart. And it persists even if you take off the stimulus which has evoked the change. So in a more precise way, it is disorder growth that is triggered by a series of events, molecular events, that are genetic events through mutations, affecting a single cell and its clonal progeny, giving rise to neoplastic cell clone, and further conferring survival and growth advantage, leading to excessive proliferation. And that proliferation, interestingly, is not dependent on the physiologic growth promoting and growth inhibitory signal, especially the growth inhibitory signals are knocked off. So that is autonomous, excessive cellular proliferation under the influence of some genetic aberration. So when you say benign tumor, it is well, that is benign. And gross and microscopic appearance of these benign tumors, they look innocent, they're homogeneous. There is no bleeding, no hemorrhage, no necrosis, no cystic change. They kind of look very localized, encapsulated, well circumscribed, do not go haywire, they do not go to any other site, and they're amenable to local surgical removal. So some of these examples are here. So what are malignancies? We can have sarcomas arising from the mesenchyme, carcinomas which have an epithelial cell origin, lymphomas, tumors of the hematolymphatic system, leukemias also, and when you do not know where it is coming from, what is the specific lineage the tumor belongs to, we can say it is a malignant neoplasm, but it does morphologically, it doesn't fit into anything, nor by immunohistochemistry we could assign them into melanoma, sarcoma, carcinoma, or hematolymphoid category. We are left with this terminology called poorly differentiated malignant neoplasm. Also, when you do an imaging, you did not find any mass anywhere, so it is either a unknown primary thing or it is something where we cannot assign a name. The sequencing or the genetic analysis has an immense role in this group of neoplasm because which chemotherapy you want to use, you have no clue. And secondly, we need to find out some targets so that we can have some drugs against this target and treat the patient. So it's a genetic disease. In essence, it is a genetic disease, an accumulation of molecular alteration in the genome of the somatic cell is the basis of cancer progression. The very first thing is the APC mutation, which is a tumor suppressor gene in the 5q in the Q arm of fifth chromosome. And when the normal epithelium, when there is a loss of APC, which is the tumor suppressor gene, that is this plastic epithelium, with an early adenoma formation is the low-grade dysplasia, leading to an intermediate or late adenoma with high-grade dysplasia. Subsequent P53 gives it to intramucosal carcinoma, and further changes in MET and the Pi3 kinase and other pathway gives it the metastatic potential. So there are certain initiating players and some propagating players. So there are few terminology which are basic, and their basis they make the foundation of molecular genetic pathology. What are chromosomes? Chromosomes are normally present within the nucleus of a cell, and they are not visible unless the cell is dividing. They are like pulley structures, or they're like thread-like structures, and thread becomes more visible when the cell undergoes dehydration in the beginning of mitosis. And you see the chromosomes, and there is a central thing which is a central, or it could be eccentric depending on the type of chromosome we are dealing with, and that is a centromere, and you have a P arm and a Q arm, a petite and short arm and a Q arm. And genes are segments of DNA that are present on the surface of the chromosome, and DNA stretches when they combine to make the gene. So the first is the chromosome, the largest one, then the gene, and the smallest structure is the DNA. So gene is not larger than DNA. Sometimes you think like gene is not larger than DNA, or DNA is larger. They can be of the same size, or gene can be larger. So they are like, this line is very important. The chromosome is the largest, which contains gene, and genes contain it. A human cell contains approximately 30,000 genes. And the traits we inherit from our parents are determined by one or many genes, and a single gene can express one or more traits. This is important. And this is the important thing in oncology. One or many genes can give rise to one trait, and a single gene can give rise to many traits, like one syndrome with one defect, but there can be phenotypic expression in different organs at different period of time. So if you say there's a cell nucleus with a nucleolus, and there are chromosomes with DNA, and the DNA have a double helix structure with the inside part are the nitrogenous bases. These are the adenine and guanine are the purines, and cytosine and thymine are the pyrimidine bases. So C makes triple bond with G, and A makes double bond with T. And we have, and this backbone is formed by the sugar and phosphate. So before even going further, we need to know what are we talk like genome sequencing, exome, transcription, what are those? So genetic is the determination of the entire DNA sequence, and that is inherited. And when we say genome, it is the whole DNA sequence. The genetic is a study to determine the DNA sequence that is inherited, whereas genome is the whole genome sequence. Exome is the whole genome, the whole coding DNA sequence. That is the DNA which is going to make mRNA. So that is exome is equal to multiple exons, which are the coding region. And similarly, intron are the non-coding region that is sliced out of the pre-mRNA before they export from the nucleus. And what is proteome? It is all potentially expressed proteins encoded by a genome, obviously from the exome to mRNA. Not all mRNA they transfer into a protein. Then peptidome is the protein fragments secreted by the tumor into the circulation. And what are transcriptomes? That is a set of all RNA molecules starting from the mRNA, which is formed in the nucleus and comes to the cytoplasm, ribosomal RNA on the surface of, which makes the ribosome on the surface of rough endoplasmic reticulum, transfer RNA carrying the amino acids, microRNAs, and other non-coding RNAs or ncRNAs are. And miRNAs and ncRNA play an important role in carcinogenesis. So this is what we have a big, this is a 5 prime and the 3 prime, this is 3 prime and 5 prime of DNA, a double helix, which is red ones are the non-coding and the green one and the coding are the exons. So there is intronic splicing, so the exons they combine and form the mRNA, and that is the central dogma of molecular biology. What do the miRNAs or microRNAs do? They're responsible for cellular differentiation, so a particular cell type of expression of certain type of mRNA, whereas downregulation of certain type of mRNA, and vice versa. So how we identify tumors? When you see a tumor at an unknown site, if you have a tumor, we are thinking it is coming from breast, and it has a upregulation of the best-rated gene or miRNA and downregulation of the genes which are normally not involved in genesis and differentiation of the mammary gland, we kind of think it is mRNA, but the things are not very clear. Things are evolving in mRNA. It has limited diagnostic utility. It has use in therapy, and they are tissue specific. So, with the sequencing of human genome in 2003, there is a new paradigm shift in medicine, especially in the cancer medicine, to have a gene-based knowledge combined with healthcare, evolving into personalized and further into precision medicine. So we have 3 million DNA base pair with 30,000 genes, 500,000 proteins that characterize the human genome. As I said, the genomic instability is a hallmark of cancer, and this mutation can be recurrent, which is detected across multiple patients with the same disease, or it is very, very private to a single patient. Like uh, there are two people with HER2 amplification, or there are two people with um, adenocarcinoma of the lungs, but one having a ROS and other was a MET exon 14 skip mutation, so that is private to those patients. And this information within those genome of cancer cells help us to classify the tumor, to treat the tumor, finding out the therapeutic option, predict therapy that is ineffectiveness, a person having an anti-EGFR. If there is a EGFR mutation, we are giving anti-EGFR therapy. But you haven't tested the person for KRAS, NRAS, BRAF, and PIK3. So that this patient is going to response is going to be ineffective. So you can predict it and prognosticate depending on the type of mutation and rearrangement. So the mutational landscape of an individual's cancer, it informs us how we can decide and go about the treatment based on the novel targeted therapy. One thing I want to clear here, what is a mutation? It may be a point mutation, or it may be a translocation, insertion, inversion, deletion, or isochromatic information. They come into the terminology of mutation. So the challenges for a better cancer treatment require genetic analysis. Why? The differences in the genotype require personalized treatment strategies. And so we need to know what drives the tumor growth and what targeted therapy, if any, can be used for the patient. So from a symptom-based blockbuster approach of treating a patient based on the diagnosis with some variables like growth, microscopy, clinical, and imaging and other techniques, which has a lot of side effects, and it has a one-size-fits-all type of approach based on radiation, surgery, chemotherapy, and all. We are moving towards a genetic-based prospective care based on the molecular repertoire of the patient. We can stratify the patients molecularly into different risk groups with a less or no adverse effect, and we can monitor the disease in a more we can monitor even after chemotherapy, but the monitoring here is more at a molecular level, looking at the cell-free DNA or looking at the transcript. We can monitor how much of the disease there is, a minimal residual disease. So in that way, you can offer the right treatment for the right person at the right time with the right dose for a better outcome and improved quality of life in our cancer. So there are certain facts you all need to know about the human genome. The number of cancer-causing genes, they have between 300 to 600. So we will know like why there are so many. The NGS plates are kind of arranged in that way, the gene chips. And the protein coding gene, they comprise 1% of genome, 99% is rest. The number of genes in a genome is 30,000. A number of gene coding for protein is 20,000. Number of base pair in a genome is 3 billion. And how many cells you need for the tumor to get noticed, you can see a swelling is 10 to the power of 9 or 1 gram. So there are 4 classes of regulatory genes which are the main targets for cancer-causing mutation. And those are proto-oncogene, tumor suppressor gene, DNA repair gene, apoptosis regulating gene. So when you say proto-oncogene, so they are responsible for the normal cell growth and proliferation. So if there is a gain of function mutation in the proto-oncogene like PDGFRA or TKIT in gastrointestinal stromal tumor, or tumor suppressor gene normally which knocks up the growth stimulating signal and gets the cell into a normal resting stage. If that is deleted or that is inactivated, like P10 deletion or inactivation by hypermethylation of the promoter region of a gene, or there is BRCA mutation, there is loss of function mutation in the tumor. DNA repair gene. DNA repair is a good process, but if the DNA repair process is kind of disorganized or dysregulated, there is a loss of function mutation. And apoptotic gene, there may be gain or loss, which may lead to a mutated phenotype and genomic instability, which is the hallmark of cancer. Okay, let's see the answer. So it has been known for almost a century that certain viruses can cause malignant transformation. And the responsible genetic agent identified in the viral genome, which is called the V-onc, and it is found that homologous of this V-onc existed in the human. So these normal human genes, genetic elements, are given the name proto-oncogene. And one of the first is the SRC, which is identified in the avian route sarcoma virus that has been studied as a transforming virus decades earlier. In the most cancers, mutations or aberrant expression of oncogene have been identified. So the answer would be, so what are the accelerator and the brake mechanism of cancer, or you can say passenger and driver mechanism of cancer. So the tumor suppressor gene, when they're unmuted, they kind of make breaks in the tumor region, and they're mutated throughout the gene, or the mutations can be truncation, so the protein is not formed, or that mutation may enable, it may not be a truncation mutation, it may not totally form the protein, or the protein forms is defective. So, and it has to affect both the alleles to be expressed, because most of the tumor suppressor genes, they are in a recessive format. So if there is a loss of heterozygosity, in a heterozygous way, they cannot express. It has to be homozygous. That means there is to be affecting a loss of both the alleles to be expressed by tumor suppressor gene. Whereas in oncogenes, if we have this accelerator mechanism, which is continuously pressed, so because it keep on working, and the mutation in the hotspots of this accelerated gene is responsible for cancer, and mostly they are missense mutations, they are dominant, and they can be expressed in a heterozygous state. So you have the pictures of accelerator and the brake. So one question here, which of the following is true with regard to the genetic mutations in cancer? Gain of function mutations are generally dominant at the cellular level. Loss of function mutations are generally recessive at the cellular level. 90% of germline mutations in the familial cancer syndrome are in the tumor suppressor gene, or all of the above. The answer is 90% of germline mutations in familial cancer syndrome are in the tumor suppressor gene. It is thought that any dominant oncogene mutations likely cause fetal demise and therefore do not carry family in cancer syndrome. So because so here there is a little hint. So when you are saying genetic mutation, if it is a gain of function mutation at the genetic level, I mean not in the somatic cell, in a cell which is getting inherited, that leads to fetal demise. So that is really not transcribed into a tumor, kind of reflected in a tumor. So, and 90% of the germline mutations in the familial cancer syndrome are in the tumor suppressor gene. They are not in the oncogene. So the first one is definitely not correct. Second is the loss of function, that is mostly for the somatic loss of function that is in the tumor cell, not in the rest of the body. And so it is not of the gain of function mutation in the oncogene in a somatic cell is something you see in most often in the proto-oncogene, and loss of function, the tumor suppressor gene in a somatic cell. But if we're really talking about the cancer which are inherited, so in an inheritance level, this loss of function mutation, most of the mutations are in the tumor suppressor gene and not in the oncogene. So the best answer would be C, and the next best would be B, not A. Is it clear? So now talking about the somatic mutations in cancer. So we have passenger and the driver mutations. So what are the drivers? The drivers, as soon as the driver gets into the bus or the car, the bus starts. But the passengers, they come and sit inside the car. The drivers, they contribute to the development of the malignant picture in the tumor or the malignant phenotype. The initiating driver mutation is the one that starts with all, but one is not enough. So you need multiple drivers, like in uh, non-small cell lung cancer, we have EGFR, ALK, ROS, MET, BRAF, HER2, MET, and RET, and other many genes. But there are some passengers also. The passengers, they have no effect on the phenotype. They acquired en route or before, during, or after the driver mutation. And sometimes the passenger gives advantage to the driver to work, but alone they may not work and affect and knocking off the passenger effect, if they are present in the tumor, is not going to eradicate the complete tumor because the driver has to be addressed. So our always a target in cancer to find out the driver mutation, which drives the growth of the tumor. So which of the following is true about somatic mutations in cancer? Driver mutations outnumber the passenger mutations during oncogenesis. Driver mutations always involve the oncogenes. Passenger mutations may arise only after the initiating driver mutation is established. Driver mutations mostly fall in the protein coding region of the genome. So the driver mutations, they instruct the cell to function as an autonomous clone. It is causally implicated in oncogenesis that is responsible for the cancer. These mutations may contribute to the development of malignant phenotype, and there are about 300 to 600 protein coding genes which are the driver, and that can involve both the oncogene as well as the tumor suppressor gene, like TP53, which is present like universally, that is a tumor suppressor gene. So driver mutation doesn't necessarily involve the oncogene, it can also involve the tumor suppressor gene. So there can be a gain or a loss of action in that. Passenger mutations are acquired by the clone before the first driver aroused or during or after its subsequent transformation. So the correct answer in this would be, what is the correct answer? C is not correct. And driver mutation that does not necessarily fall into protein coding. It could be intronic drivers, and they outnumber the passengers. No, the best answer would be usually the passenger mutations are way more than the driver mutation. If you sequence a tumor, you see probably 2 or 3 drivers and hundreds of passengers. So the best answer would be driver mutations are always, that is the key to go for oncogenesis. You may have or may not have a phenotypic expression, but that is something which is responsible for oncogenesis. Is it clear? Okay, so coming to the origin of species by Dr. Charles Darwin. So the cancer cells will follow a Darwinian theory to survive. All tumor cells are progeny or clonal, they are identical. There is a natural selection process, the survival of the fittest subclone. So within a tumor, you see tumor heterogeneity, multiple clones showing different genomic repertoire, and the or the genomic armamentarium, or you can say like the genomic structure or the color and whatever, whichever way you want to kind of present them, the clone with the more aggressive genes is going to metastasize. So there is a natural selection process within the clone subclone, and which is called clonal diversity. So the cancer cells that develop due to a stepwise accumulation of complementary mutations over a period of time. So the normal cell with a carcinogen-induced mutation leads to an initiated precursor with stem cell-like properties because they kind of go back to the primitive stem cell. There are certain mutations which affect the genomic integrity, leading to precursor with a mutator phenotype. With additional drivers come accumulations of driver, a number of passenger mutations, but the foundation of cancer cell is laid out here. With additional mutations and emergence of subclone which are in different color, the tumor becomes genetically heterogeneous, and where we see the tumor and diagnose it. So we have an initiating mutation, acquisition of more and more genomic instability, acquisition of hallmark mutation, which is going to forward the foundation of the tumor cell with further genetic evolution and spread. So there are 8 characteristics which are fundamental changes in cell physiology, which are considered to be the hallmarks of cancer, which are all here. I'll go one by one. So when you talk about the cancer, we talk about receptor tyrosine kinase mediated pathway, which is most frequently mutated in tumor cells. Why? We have a receptor, and most of these receptor, as you know, there's an extracellular domain, intracellular domain, and transmembrane domain. The growth factor gets attached here, sends the signal, which get phosphorylated, and then you kind of the inactive ones gets activated by phosphorylation. So whether you have an ATP or GTP, or so that gets converted, and that further activates the pathway. So these mutations in the receptors. So I have given one example of EGFR, but it can be anything. And what happens? This mutation in the receptor lead to constitutive tyrosine kinase activity. So they're kind of increase in the tyrosine kinase activity in an exponential way in response to the growth factor. But if the growth factor receptor is mutated, and even one growth factor is attached here, it gives rise to an exponential action. Like there's a burst of growth factor activity because the receptor tyrosine kinase domain, which is usually present juxtaposed to the membrane, below the membrane, attached to the intracellular domain of the receptor. So like in EGFR mutation in adenocarcinoma of lung, or amplification as receptor, HER2 in CA breast and bladder, and ALK in adenocarcinoma of the lung. RAS mutation, as you say, is the most common mutation in the human cancer, which is a K, H, or N RAS. So what happens? This receptor tyrosine kinase, the KD area, the kinase domain, is attached to the inactive RAS, which gets activated to active RAS by acquiring a phosphate from GTP. And GAP, what it does, it will inactivate by hydrolysis of GTP. Then it further activates the active RAS goes, and they either go through the Pi3 kinase pathway or a RAF and MAP kinase. So we say RAS, RAF, and MAP, and BRAF is one of them, which ultimately they focus into the activation of transcription factor. And so inside in the body, we have inherent GT page, which basically make the active RAS into inactive RAS, and by hydrolysis of the GTP. And there is another pathway where there's a Pi3 kinase pathway, which has a catalytic and regulatory subunit. So Pi3 kinase mutation, PIK3CA mutation, um, is in hormone positive breast cancers, and metastatic breast cancer have a targetable drug. And similarly, mTOR, which is in the same pathway through the AKT, which is in metastatic RCCs and metastatic breast cancer, particularly the ER positive breast cancer, it is upregulated through the upregulation of Pi3, and ultimately it gets into the cell cycle proliferation. And the activating transcription factors are MYC and MAP, which are present within the nucleus. So this anti-EGFR molecule, when you talk about anti-EGFR in metastatic colorectal carcinoma, works only in the setting of unmuted or wild type RAS. Why? Because if I get back to the other picture, you know the EGFR is here and the downstream is the RAS. Suppose there is no RAS mutation, you give an EGFR blocker. It like suppose in lung cancer, you give like osimertinib, which is going to, or cetuximab, or any kind of drug which is a good EGFR blocker, it's going to block the EGFR and it works. If there is a downstream mutation in the RAS, it is not a wild type RAS, it's a mutated RAS, then this is not going to work. If there is an mTOR amplification, and because normally what happens, P10 controls the action of Pi3 kinase. If P10 is deleted, or there is loss of heterozygosity for P10, and it is tumor suppressor gene, normally it doesn't work, then this pathway gets activated, leading to cell cycle proliferation, even if you are giving one target molecule. So this line is important, like anti-EGFR antibodies does not work unless you have an unmuted RAS or a RAP pathway. Similarly, what are the non-tyrosine kinase mediated pathways, like a JAK-STAT pathway your myeloproliferative neoplasm, or you have. So what happens? These receptors or these things, I mean, whenever there is a 922 translocation, like in chronic myelogenous leukemia, that leads to a 22q deletion. So this is basically what a 22q deletion, because this part has gone, gets attached here. You see this blue thing is here and this thing is here. So this is the 22 deletion. And so this BCR locus, which is in the 22nd chromosome, and the Abelson locus in the 22nd chromosome, which is the newly formed 22q minus, or the Philadelphia chromosome, makes a hybrid. And here the oncogene is Abelson, and the BCR is the breakpoint cluster, which is next to it. And they have a tyrosine kinase activity. They do not need a specific receptor. So this is a great example of non-receptor mediated tyrosine kinase pathway activity, which activates the various growth factors. We can have kinase domain mutations, that is the mutations in the tyrosine kinase area are BCR-able. So for that, over a period of time, the patient does not respond to imatinib. So they need more drugs like dasatinib, denotinib, or ponatinib if there is a failure of response to imatinib. And we have the test to diagnose PCR-based techniques to know whether there is a KD mutation in those patients or not, because why the patient is not responding. And so the Philadelphia, there's the first one to be discovered as a paradigm shift in cancer in 1959, 73, and 2001. And these are the Nobel laureate. We kind of put forward the foundation of molecular medicine by Dr. Noel, Underwood, Rowley, and the group, Sawyers, Drucas, and Leiden, who developed the kinase inhibitors, imatinib, and which was the first one to be used in oncology, the first TKI, and is FDA approved. So all the downstream signals, they ultimately converge into nuclear transcription factor upregulation, or I can say nuclear transcription factor stimulation. And inappropriate and continuous stimulation lead to nuclear transcription factor to drive growth promoting gene. And finally, what are those factors? MYC, MIP, Jun, and FUS. So they stimulate, again, those translocation like 814. Suppose we take an example of Burkitt, normal 8, normal 14. So if you go to the 8th chromosome and see, you have the 14th chromosome has the immunoglobulin heavy chain gene. And the MYC is at the terminal end of 8th chromosome in the long arm. So when there is a translocation, so this is normally under the influence of the MYC is under the influence of a lot of growth inhibitory genes next to it, so it doesn't get expressed in normal cell. But when this gets translocated or rearranged with the immunoglobulin heavy chain, which gives it a survival or a proliferative advantage with increased MYC expression, and that increases the expression of pro-growth gene, which leads to cyclin D-mediated cell cycle progression from like a G0 to a G1 and further to S and a G2 substage, then protein synthesis and reprogramming of genes in that manner. Then what are the cyclins and the cyclin-dependent kinases? They are the crucial for driving the cell through the cell cycle. And we have the CDK inhibitor and the cyclin inhibitors, which knocks off the action of all this protein. If I talk about P16 we say like P16INK4 because it normally inhibits the action of cyclin and cyclin-dependent kinase 4. So when there is so it inhibits the promotion of G1 to S. When P16 is mutated, being a tumor suppressor gene, there is promotion of the cells. The tumor cells, they could change from a G1 to the S, and further they get into the mitosis. And this is for the RB from G1 to S. And so these are the few things you need to, and P21 is G2 to M. So these are the areas where you need to know, and in the greater details I used to take in my class, but we have less time. So the CDKs, they do work. But definitely the P16 you need to remember, and the RB. So the cyclin responsible for facilitating the progress of cell into and through mitosis. I'll cheat here. The answer would be B, because if I go here, cyclin B, which is this one, this is important because if you knock off cyclin B, a cell cannot enter the mitosis. You can knock up this one, you can knock up this, but this is one, the cyclin B, which works with cyclin-dependent kinase 1. You have to knock up either of them or one of them in order to halt the cancer cell to grow. So because they kind of work at the G2 to M transition. And these are the CDK inhibitors like P21, P27, and 57. They work on the cyclin B level. And the second one is which of the following is necessary for G1 to S phase transition. So G1 to S phase transition is cyclin E and A accumulation. If you see the G1 to S here, so you need to have accumulation of E and S to work. So if you work on that, then it won't progress from G1 to S. So these are some of the targeted therapies we see when there is a growth factor related gene thing, and the receptor for those growth factors are the proto-oncogene, which were mostly, if you see here clearly, they're overexpressed or amplified. That is the gain of function mutations like PDGF beta for astrocytomas. You have hepatocyte growth factor for hepatocellular carcinoma and thyroid tumors. You have red point mutation for MEN, KIT gain of function. So most of these are gain of function, either through overexpression, amplification, translocation, or point mutations. So that most of them, if you see it very clearly, these are not truncation mutations. That is one point I want to highlight here. These mutations are gain in function mutations because it's involving a proto-oncogene, and these are not truncations. Either they're overexpressed, they're amplified, they are mutated, or there is a fusion because of translocations, or there is a additional chromosomal matter material is attached to that area coming from another gene. So these are all non-truncated mutations activating in the tumors. Then coming to this evading tumor growth suppressor, which is something very important. So the tumor suppression, the RB gene is the first one. Retinoblastoma gene is the first ever tumor suppressor gene to be identified. It has an inhibitory effect on the cell growth, and the protein or a gene that is associated with suppression of any of the various hallmarks of cancer. Bracha gene is another tumor suppressor gene, which prevents genomic instability by repairing defects in homologous recombination. Whenever there is a DNA repair defect, regenerative damage, Bracha seals it up. So it is helpful in the repair of the homologous set of chromosome, which is called the homologous recombination technology or the methodology or the process. And when BRCA is knocked up, you see defect in the homologous recombination leading to genomic instability and cancer. So what is RB? RB is the governor of all cell cycle. It is inactivated in most human cancer. So let's see. When a growth factor, and the left side is the normal and the right side it is abnormal. So growth factor that stimulates the CDK inhibitors, like growth factor inhibitors like TGF beta, and so cell is growing in a normal way. So we have growth factor inhibitors like P53 transforming growth factor beta. They stimulate the cyclin dependent kinase inhibitors like the INK4A or P16, which further inactivates cyclin D and E. So that leads to hypophosphorylation. The phosphate 3 minus group is off. So when that is off, the RB is non-phosphorylated or hypophosphorylated. That is actually hypo, not non. It is hypophosphorylated. So that leads to, if you see here, the E2F will work with RB, and this DNA histone acetylase, which are present within the nucleus as nucleoprotein, and histone methyl transparent, they get attached to this pocket, and that leads to knocking off of the E2F elongation site, and the cell cannot get into mitosis. So there is a transcriptional block. So when there is a loss of function mutation in both the RB alleles, I'm repeating, we need to have a loss of heterozygosity either in a germline mutation of retinoblastoma, or there is a somatic mutation in small cell carcinoma, where you see small cell neuroendocrine carcinoma. We have a RB's loss of heterozygosity, loss of function mutation that leads to loss of function and of that particular gene, and gain of function. RB loss is expressed as the gain of function mutations in cyclin D and cyclin dependent kinase leading to transcriptional activation. If you see here, if RB is in a hypophosphorylated state, it makes a layer over the E2F elongation factor. It doesn't allow the E2F elongation factor to work with other factors like histone disacetylase and methyl transferase, and because they also inhibit. So there are three layers. One is methyl transferase, one is deacetylase, and one is your hypophosphatemia RB. So all three, they kind of seal up the structure of E2F on all the sides and do not let it go for further transcription. So we have like brain tumors. We have a histone deacetylase or the HACT type of tumor. We have a methyl histone transferase group of tumor. When these tumors suppressor genes are knocked off, you see different types of glioma with different histologies. So that is the basis, because the main culprit is E2F and they want to like seal off the action of E2F, so it doesn't really work. Then comes the TP53, which is the best, the guardian of the genome, is the most commonly mutated gene in cancer. So whenever we have a radiation, carcinogen, or mutagen, the cells with normal cell, what happens? There is DNA damage. So whenever there is DNA damage or there is some oncogenic stress or hypoxia, P53 automatically accumulate and they try to bind to the DNA. So this oncogenic stress, in addition, also stimulates something like or hypoxic thing. So that what it does, it accumulates a lot of TP53. So what it does a transcription dependent and independent effect on the target. It stimulates the Bax gene, which is an apoptotic gene. So the cell will undergo apoptosis directly, or it will try this to rectify the cell, the damage. So either the cell will directly get into senescence if it is an old cell, or if it is a young cell, it stimulates the P21, which inhibits the CDK, so there is a G1 arrest, or it stimulates the GAD45, which is the DNA repair gene, leading to a successful repair. So either way, it is either going to die directly or through the Bax pathway, or it is old, it stay in a senescence way and followed by apoptosis later on, or it is a young cell, it undergo in a normal situation when P53 is intact, it inhibits the CDK through P21 and having a G1 arrest, or repair by GAD45, which is a DNA repair machinery pathway. Then if you have a cell with a mutation or loss of P53 with a DNA damage, there is P53 dependent genes, they are not activated. So there is no activation of all these pathway downstream P53. So there is no cell cycle arrest, there is no DNA repair, and no senescence and no apoptosis, leading to mutant cells with expansion and additional mutations. So we know P53 loss of function is seen in Lee-Fromenich syndrome, and so when the P53, there's a loss of function mutation, which is somatic mutations in the germline way, that is called Lee-Fromenich syndrome, and there is something called a gain of function mutation in MDM. If there is amplification of MDM2, which we see in a lot of tumor. One of them is a dedifferentiated liposarcoma, and also in liposarcoma, the well-differentiated one as well. So gain of function mutation of MDM2, which is an oncogene, which is amplified, and we do it in a FISH technique similar to FISH, that degrades TP53, leading to a sarcoma. And functional inactivation of TP53 can happen with HPV-E6, and you see it in carcinoma of the cervix. So germline mutation germ cell tumors have an intact P53. This makes them highly susceptible to genotoxic stress like cisplatin. So if you're giving the cells, the tumor cells, the germ cell tumor, your seminoma or embryonal carcinoma, they usually have an intact P53. It is not mutated. So these cells they are highly susceptible to cisplatin. DNA damage leads to hyperactive P53 and apoptosis. So this is somewhere. And there are few other tumor suppressor gene, which is ATM, ATR phosphorylate, and they liberate p53 and MDM amplification, which are also kind of work hand in hand with p53 knocking off its effect and further cell proliferation. Then effect of TP53 mutation, the role of p53 cancer-related process, which I have already shown you. So if either it is completely lost or it is high, whenever we see, there is one thing, in any organ, you stain the tumor, any tumor you stain, or any normal organ you do p53 staining, you see some kind of staining. But if you see it is completely gone or it is extremely highly expressed, that means either it is a null phenotype mutation or overexpression, or there is a mutation where the gene is lost but the protein is expressed. So, and you know the loss of heterozygosity is responsible for this thing. So whenever we have a truncating TP53 mutation, there is one on the left, one on the right. This is important concept to remember. Whenever there is a truncating mutation there, that means the protein is there but not completely expressed. So there is a loss of heterozygosity. So there is lack of transcription and translation, leading to absent TP53 because the protein is not there. So you would not see the TP53. Whereas there is a point mutation leading to abnormal p53 sequestered in the nucleus. So what you see is a lot of p53 signal within the tumor. So either the malfunctioning abnormal p53 or a complete absent non-phenotype of p53 that leads to loss of control over few processes like DNA repair, apoptosis, senescence, and cell cycle regulation. The second one is deregulating cellular energetics. So I'll talk a few things about Wartburg effect. Normally what happens, the normal differentiated cells, they require oxygen for to undergo the Krebs cycle, and they release 36 to 38 mole of ATP per glucose in oxidative phosphorylation within the mitochondria. But whenever there is anaerobic glycolysis, the final product is lactate with 2 mole of ATP. So what happens in a proliferative tissue or in a tumor tissue, with or without oxygen, it forms pyruvate. And among that, 85% of pyruvate that gets into lactate and 5% that goes into the mitochondria, even in the absence of oxygen. This is something, a beauty you need to remember. Even in the absence of oxygen, in an anaerobic way, with a less amount of oxygen supply to the tumor cell, 5% do go into the mitochondria and produce some energy, and the mole is at least 2 more mole of ATP per mole of glucose, and that is called aerobic glycolysis in tumor cells, and this is called the Wartburg effect. And the tumor cells, they kind of survive on that. So this fine-tuning of metabolism for biosynthesis, the tumor cells, they try to fine-tune the biosynthesis of nucleic acids, lipids, and protein rather than forming ATP. It doesn't kind of waste a lot of time in making ATP. Rather, it try to fine-tune the process so that more and more nucleic acid, lipids, and protein can be formed so that the tumor can grow. So here the same pathways are there. One thing I just want to highlight, if you get into the Tca cycle in the mitochondria, these two enzymes are important. One is the succinate dehydrogenase, and you have fumarate hydratase, an enzyme of tricarboxylic acid cycle. So those are normally okay. If there is an hypoxia going on, the tumor cell is creating hypoxia within the cell, and in the absence of these enzymes, so that leads to liberation of a lot of this thing, the HIF, which is hypoxia inducible factor, that further stimulates oncogenesis. So in nutshell, you need to know that. And similarly, you have this isocitrate dehydrogenase in brain tumor, IDH. We say IDH mutations. So this IDH normally makes alpha ketoglutarate. A mutation will either make a lot of it will knock off this process and leading to accumulation of IDH and cancer. Then deregulating cellular energetics to Warburg effect. In oncogenes, they promote, and tumor suppressor genes, they inhibit. So tumor suppressor gene, they normally also work by inhibiting Warburg effect, do not make the cell to make more and more ATP, and so there is no generation of intermediates for proteins nucleic acid synthesis. Whereas in a tumor cell, there is always that process is going on because there is a glucose transporter which is independent of this thing. If you see a quiescent cell, there's nothing happening. The cell is kind of making 36 ATP and is happy. But here, if you see, is a proliferating cell or a tumor cell, what happens? This pathway is going on with the glucose getting inside it, and there is a and in the growth factor is also stimulating. If the tumor suppressor gene effect is knock off, what happens? These glucose they're here, they kind of rerouted instead of getting into the mitochondria, they get rerouted towards making nucleic acids, protein, and amino acids. And for subsequent tumor cell growth, and very little will get into the mitochondria to form two more ATP. So what is the basis of Warburg effect when you use this 18 FDG PET scan? So it helps in glucose metabolism. This is a tumor with pre-therapy. There are two FGP PET positive nodule in the liver. The patient underwent therapy. So what you see, there is excretion in the bladder and in the kidney. This is the excretion signal, and there is nothing inside the liver. That means it has undergone metabolism and is getting excreted. So that is the basis of FDG PET CT scan, and it uses the Warburg effect as the principle. Then registering cell death. Registering cell death or evasion of apoptosis is another mechanism of malignancy. So intrinsically in the mitochondria, the pathway is more frequently is visible in cancer. BCL2, as you know, is overexpressed in many B-cell lymphomas and leukemia. Then MCL1 induces resistance to chemotherapy, so you can have drugs for BCL2 which can be used. So in the intrinsic pathway, whenever there is a DNA damage that leads to P53 response and leads to a BAX activation, leading to this upregulation of BCL2, BCLXL, and MCL1, and for the process going on. So whenever there is increase in the decrease in the anti-apoptotic factor, increase in the apoptotic signal, that leads to cell death. So this is the way, and this is the extrinsic we know which go to the FAS and FAS ligand pathway. But these are the three factors: BCL2, BCLXL, and MCL1. They are the anti-apoptotic factor, and this is the pro-apoptotic factor. So there is an imbalance, or there is upregulation of BCL2 or MCL1, leading to evasion of apoptosis and cancer. Then comes the enabling replicative immortality. The cells have discovered the proverbial mountain of everlasting youth. So this HeLa cell line we have been using for over 60 years, the immortal life of Henrietta, and we are going to use it for more and more cancer evolution. So this enabling replicative immortality through the evasion of senescence, evasion of mitotic crisis, and a process of self-renewal by this telomerase. So in the germ cell and the cancer stem cell, the telomerase is intact, and 90% of the cancer, the telomeres are maintained due to regulation of telomerase. If the telomerase is working fine, then the telomeric shortening doesn't happen because it's a DNA polymerase, and it will keep on making the new telomeres which have undergone destruction or they're deleted or whatever happened to them. So they make the cells immortal. The telomerases do not go. Normally what happens when there is an inactive telomerase, there is shortening of telomeres with multiple in about 50 to 7 cell division, and cell is dying, destined to death by activation of P53 and checkpoint signals like ATM and ALB2. And what happens in mitotic cell when these pathways are knocked off, that leads to a bridge fusion breaking mechanism. That is, you have a dicentric. There are one chromosome with two centromeres, because these dicentric chromosomes happen to, in order to the telomerases are making more and more segments of nucleic acid DNAs, and they kind of lump on that existing chromosome, which further undergo mitosis and make the cells alive. When there is too much dicentric catastrophe of dicentric chromosome-related mitotic catastrophe that leads to reactivation and reactivation of telomerases, leading to cancer. Then cancer stem cell. Cancer stem cells, they have a self-renewal. Each time a stem cell divides, one of the progenies retain the property of the stem cell. And difficult to eradicate them because they have a low proliferative activity. They develop multi-drug resistance, and high-dose chemotherapy is usually response needed to knock off those self-renewing population of stem cells. Then comes the neoangiogenesis or neovascularization, because tumor is the process of anti and pro-angiogenic factors. P53 inhibits vessel formation through inhibiting VGF and thrombospondin, whereas hypoxia leads to HIF1 and more VEGF expression and fibroblast-derived growth factors. Similarly, mutated P53 promotes angiogenesis. So when there is a gain-of-function mutation in the RAS, RAF, and MAP kinase, the MYC amplification leads to VEGF. So these are some of the pathways. So whenever there is hypoxia, in any malignant tumor, you see a lot of necrosis, and you have a survival pattern of existing cell surrounding a blood vessel. That means the cells which are surviving, they are surviving next to the blood vessel and trying to make new blood vessel through the hypoxia-inducible factors so that they can grow in a snake-like manner throughout the body. Then anti-VEGF therapy has modest benefit, and these are the some drugs which are used for anti-VEGF. And the action, how they work, these anti-VEGF drugs, they prevent the growth of vascular endothelial, they prevent the action of vascular endothelial growth factor, so the tumor cell cannot grow. Then comes the genomic instability or the mutator phenotype. Genomic instability defects in the DNA repair mechanism. We have a double-stranded DNA here. So you can have, if I come to various tumors, these are the various genes you need to, at least 4 or 5 genes you need to remember. One is the BRCA1 and 2, PALB2, ATM, and CHEK. These are the genes which are normally they're responsible to repair the DNA. So genetic aberrations in these genes are mutations leading to one defective copy and the other one is normal. When 2 defective copies are inherited, or the tumor has both the defective copies in a somatic cell. In a germline way, you have one defective copy coming from the mom and one defective copy coming from the dad, or in a somatic way, within the tumor, there is homologous recombination defect or DNA repair defect because of the deficiency of both the genes among all these 4 or 5 genes that leads to a defect in the double strand break repair and leading to various types of tumor and BRCA-related breast, ovarian, pancreatic, prostate cancer as a thing. And PARP inhibitor, what it does, PARP inhibitor, it will help because if in a tumor cell, the tumor cells develop a self-renewal technique. Even if the BRCA is gone, so the tumor cells, what happened, the suppression effect is gone. If there is a defect, it gets completely repaired in the tumor cell by an adduct which gets attached to the tumor cell. That adduct is going to be inhibited by PARP inhibitor. So this PARP is the adduct. PARP is the adduct, or it is again another polymerase, nucleic acid polymerase, and that helps that particular area, this DNA repair the breaks to get filled up in the tumor and make the tumor cell to proliferate and grow. So if you give this PARP inhibitor, platinum salts, they inhibit those areas, leading to the defect will still stay there and the cell is going to die. Then what is synthetic lethality? This is something we need to know. There's a functional BRCA. This is normal on the left and this is abnormal on the right. So whenever there is a DNA damage, inducing the single strand break, what happens? In a functional BRCA, and this is dysfunctional. So this functional BRCA is what happened. Normally, the DNA undergo repair because the BRCA is competent here, how the DNA gets repaired. So this PARP1, what it does the persistent accumulation of single strand breaks. This BRCA will accumulate, and then it forms a double strand break and collapse of the replication fork. So there is another factor called XRCC1. The XRCC is in in relation with the PARP they combine. So up to this step, it kind of try to make a cancer. So PARP1 and XRCC1, they combinedly they work, and what they do, they try to make short patch repair and long patch repair. So there is error-free homologous recombination. When and on the other side, they keep on accumulating a single and the double strands, making a fork. And if BRCA is competent, the repair process is complete, neat, and the cell will survive. Whenever there is a DNA damage induced when a BRCA is dysfunctional, it's incompetent. So even if this XRCC is there and PAR1 is there, they make this short patch and the pair, but there is you cannot get an error-free recombination. This pathway will try to make an error-free recombination. However, there is no contribution from this side, leading to a homologous recombination deficiency, leading to patient failed DNA repair. So if you give in this, how it works, if you give a PARP inhibitor, whenever there is a BRCA incompetency, it is not going to do this neat pathway. So this PARP inhibitor will work on PARP, and these two are completely separate from each other, and then this will be activated. So there will be an error-free homologous recombination, and the normal cell will stay and the tumor cell will die. So then there is something called activating invasion and metastasis. So this is you all know from Robbins. Then avoiding immune destruction. So avoiding immune destruction, normally the CD8 positive, the cytotoxic T cells are responsible for tumor cell immunity. So there are certain ways the tumor cell antigens which are present on the surface of the tumor cell, how they can make a cloud. Normally the tumor cells express PDL1, and the immune cells, they have PD1 on the surface. And so what happens is PDL1 and the PD1, they make the program death molecule 1, they make a cloud, and that cloud is over the tumor cell, which escapes the normal CD8 positive T cells to attack on the tumor cell, because we cannot identify those cells because it's already covered by those clouds. So if you give a PD1 blockers like nivolumab or pembrolizumab, or L1 blocker like duratrizumab, so that is, or a CTLA4 blocker, they are going to work on this molecule. So any tumor with a high PD1 or PD-L1 blocker, they can be dealt with this molecule. So this is the essence of releasing the Brex phenomena or the immune checkpoint blockade. And then coming to the certain tumors with the genetic abnormality, there are like different types. So we'll switch into the simplified workflow in a clinical molecular diagnostic setting. So what do? We receive the specimen, the pathologists review the specimen, then the process comes of extraction of DNA and RNA. We set up the assay. We are going for a single gene assay or multi-gene assay, or it is an in situ process where you can see the cell and see the aberration, or it is an non-in situ or in vitro assay. So I'll not go about this case. So I'll go about what are the things we do. Whenever we are doing the various methodology we do, we either see the entire chromosome by karyotyping or cytogenetics, or we look into the signal by doing fluorescent in situ hybridization, the cell is intact, or go for a qPCR or a real-time quantitative PCR or sequencing. So this is the normal karyotype. We do not know the aberration. You just laid out of the chromosome and look into it. But the problem with this technique is it needs fresh tissue. You cannot use paraffin-embedded tissue. We need the media. The cell harvesting should be good, and it has a longer turnaround time. And it can only, if you have a microdeletion, very microscopic level of aberration, you cannot find in cytogenetics. It is a cumbersome exercise. It takes a lot of time. And it is based on banding techniques from centromere to the telomere, and from that you assign a particular band. And it takes about 5 to 7 days. So it's a time-consuming process, though it is a gold standard. So this is the normal karyogram on the left, and the right you have a loss of this 22q, the 22q deletion, or a Philadelphia, and there is a gain in the 9q. So that is what we have, the Philadelphia chromosome. So it basically uses the structure of DNA, which is double-stranded. It has A, T, and CG. Double helix, we know what is Chargaff's rule. In a sample of double-stranded DNA, amount of A equal to T and C equals to G, or that is AT and CG. And the amount of purines is always equal to the amount of pyrimidine. And it has a parallel or a sense and an anti-sense or an anti-parallel strand. So in the genetic code, the central dogma of molecular biology, when there is DNA getting into mRNA and the protein, so that is what is, we use the few things we need to know here. We have, whenever there is a mutation, either there is a 10 terminating codon which is coming, which is, so these three you need to know, want to practice molecular pathology, is UAG, UGA, or UAA. These are the things which come with a mutation which forms a truncated protein because it stops the codon or terminates the protein. And protein always begins with AUG, which is methionine. There is a chain initiating codon. And whenever you say there is a promoter region hypermethylation, that means it's a methyl group attached to the promoter region, leading to gene silencing, and the gene cannot get transcribed. So the various techniques we use, we clone, we probe, we make endonucleases. So what are the differences between primer and probe, which is a basic fundamental question. Primers, probes, just remember one thing, probes are bigger than primer. Probes we use for in situ processes, the hybridizing processes, where the sensitivity is lower than the primer because we are looking at it. So if the aberration is not present in a higher frequency or in a higher level, you cannot diagnose it. So these are either single stranded DNA or RNA. The length is about 100 to 1000 nucleotides. They identify the sequence of interest through hybridization and when it is tagged with a fluorescent tag. Whereas primers are single-stranded DNA, the length is about 25 to 30. They have a free 3' hydroxyl group for the chain elongation by forming phosphodiester bond because they are making a phosphodiester bond with the residual DNA molecule which is present in the sample using DNA polymerase. And this is because in PCR, in the annealing step, we have a DNA polymerase, and that is meant for amplification. It is not tagged with fluorescent tag. So when you say in situ hybridization, the labeled probes can allow the detection and localization of DNA or RNA sequence in the cell of the tissue in an in situ process. And the probe is, when a fluorescent probe is used, we say it is fluorescent in situ hybridization. So we have the target, we have a bigger probe which is labeled with biotin streptavidin or streptavidin, and if it is related to an enzyme, the two process, either it is tagged to a fluorochrome dye, so you see it, or if it is attached to the reporter enzyme, there is a substrate added to it, and you get a product with a signal. So it is an interface phase for 922. In a normal cell, you have two green signals and two red signals. The green signals are the 22q11 and the red signal is 9q34. So whenever there is a fusion, you see a central yellow signal. That means the double fusion probe, and there is a fusion. The presence of fusion is a fusion probe. So you see the dual color ABL and BCR, and you see that is a defect in this disease, like in CML. Then PCR is a test tube based method for, and that is a slide based method. It's the test tube based method for amplifying a selected DNA sequence. So we synthesize millions of copies of a specific nucleotide in a few hours, which is definitely a high throughput technique. It can amplify the sequence even when the target sequence makes up less than 1 part of a million of the total initial sample. So it has a high sensitivity, high throughput, very less turnaround time as compared to the FISH technique. The steps are, you denature, you heat the DNA into 90 to 98 degree. You do not cool it very low. You cool it to 40 to 60 degrees so that both the strands do not join again. And you heat the primers so that the primers will start elongating the process with the help of DNA polymerase. And at that temperature is 72 degrees centigrade. So these temperatures are very crucial to remember. Each cycle doubles the amount of DNA, so there's an exponential increase 2 to the power of n in the number of cycles. And finally, you amplify the DNA and see the sequences separated by gel electrophoresis. So sequencing, and this DNA can be further amplified sequence by Sanger sequencing, which is the gold standard technique, or a pyrosequencing, single-based primer extension, or other techniques. So Dr. Sanger, who first got a Nobel Prize in 1958 for his determining the structure of insulin, and in 1980 again Nobel Prize for the base sequencing of nucleic acid, TCAG, and that is the first sequencing technique or the breakthrough in molecular biology so far as sequencing is concerned. So in real-time PCR, what you see in a normal PCR, in a regular PCR, you see the product at the end, but in real time, you see the product after each cycle. So you can have a cycle threshold after every cycle to know how much in every cycle it is amplifying. So that is to detect and quantify the presence of a particular nucleic acid sequence in a real-time way during the exponential phase of DNA amplification rather than a post-PCR complete analysis after 30 to 40 cycles. So then when you come to the personalized medicine genotype driven therapy, how it helps in therapy, we have this EGFR mutation, which is seen in about 15 to 20% of lung adenocarcinoma, and these are the other ones, unknown in 40%. So it involves the 18, 19, and 20, 21 exons. So we do a allele specific PCR in this technique and try to look for mutations. So in lung tumor now, in the advanced lung tumor, this EGFR, AL, cross, MATE, and BRAF along with PDL1 is mandatory either as a single gene test or a next-gen sequencing, but the oncologists prefer an next-gen sequencing. The tissue is always the issue. If you have a little bit of tissue left, so you want to use the tissue optimally without wasting much in the immunostain or anything, and give the best result for the patient so that the treatment can be started. And just to point one thing, before it was known like EGFR and ALK, they make the bulk, because the upcoming ones are the MET and BRAF now, and MET inhibitor, there is a skip mutation in the MET 14, exon 14, and there is a MET amplification. So if we have those actors and if you give a drug, they have a very good response as compared to the EGFR and the ALK inhibitor therapy. So see this allele-specific PCR or amplification refractory mutation system, which is used for EGFR. So what it does, the primer binds to the mutant allele, not to the wild allele. So it is called allele-specific PCR because only the mutant EGFR allele is going to bind to the primer. So amplification happens if the mutant allele is present. And this is the forward primer, which starts from 3' to 5', and 5' to 3' is the parallel or the reverse primer or the parallel sequence. I normally use the word parallel sequence as a reverse primer. And this forward primer has a reporter probe, which the sequence is complementary to the mutated EGFR. And you use a fluorescent tag substance to detect that. And the turnaround time is way shorter than Sanger's sequencing. So this is one report of the exon 21 L858R mutation in EGFR gene in non-small cell carcinoma. And here you see the base change. For that region, it is written like T greater than G. So, or there is a deletion. This 19 is mostly a deletion type. So you see a lot of deletions in 9. And here there are point mutations in exon 18 and 21. And it has some insertion in exon 20. Then in NGS is the high throughput DNA sequencing. So you see multiple PCRs going on. It is multiplexed, it is massive, and it is parallel. That means millions or billions of DNA strands can be sequenced parallelly, yielding substantially more throughput and minimizing the need for the fragment cloning method that we use for Sanger sequencing. Any DNA from almost any source can be used, well suited to heterogeneous DNA samples, and it has high sensitivity, less time consuming, and high throughput. In Sanger's, we essentially need very high quality DNA. Here we need average quality DNA with extreme degree of heterogeneity can be used to give a high yield. The limitations, it is error prone because there's high error rate in the sort reads because it picks up a lot of signals which has no significance, or you do not know what is the significance of that. It is a complex process. It makes the task of a de novo assembly of generated sequences, and it is expensive, but the price is kind of coming down, and these are the various, and we need a good bioinformatics signals. So what are the ways we need? We can be whole genome, which is predominantly look into the structural variants, point mutation, copy number variation. It sequences the entire genome, including exons and introns. It is very comprehensive, but it is most laborious and expensive. Whole genome sequencing, exploiting the 1% of the genome, is predominantly application for point mutation and copy number variation. Hybridize and capture the whole genome and is more of interest. Beds to interest, which is predominant application in point mutation and deletion, and PCR amplicon. Transcriptome sequencing is more like when you're looking at an expression like HER2 amplification, MET amplification, ALK rearrangement. You use this transcription RNA because you're looking at the gene fusion and the expression pattern and splice variants. Exon capture, so this is kind of an RNA sequencing, and RNA sequencing or sequencing of expressed RNA can be combined with exome capture technique for a very high yield. Like we are trying to like upgrade our system to include the RNA sequencing in addition to the DNA to have a higher yield, and not looking at the aberration, but also looking at the expression pattern and the splice variants in those tumors. Clinical application, I'm not going to go over it. I mean, almost everybody knows about it. The prevalence of various human cancer is kind of going, all these mutations are going more and more in number. So the mutational landscape of a tumor genome is showing each of the 24 chromosomes in the cancer is displayed in a circle, and the position of the various tumor subtypes of the aberrations, they're mapped into various cancers. So it could be a copy number, variation, loss or gain, point mutation, structural rearrangement, or a loss of a particular region by deletion. Then comes to a few words about liquid biopsy, which exploits the normal human body cell-free DNA in the circulation in the patients with cancer. The cfDNAs are released in the tumor way before the patient develops metastasis, and they amount to less than 1% of the total cell. The tumor cells that get into the cell-free tumor DNA into the DNA. Into the bloodstream by active secretion, cell necrosis, dropout, or apoptosis. Therefore, looking at the genetic signature in those blood-derived ctDNA or cell-free tumor DNA reflects the mutational spectrum in the tumor cell. Suppose you do not have an accessible biopsy site, the patient has undergone treatment, so you want to really know the mutation is existing or not in the tumor cells, because you cannot just wait for there's a tumor to develop, because you need 10 to the power of 9 tumor cells. So in a few cell-free DNA, though it is not a very sensitive technique, getting those cell-free DNA is extremely difficult. But for some of the tumors, specifically in lung tumor, for EGFR, cell-free DNA testing to look at the mutation pattern is recommended by NCCN. And ctDNA has a somatic mutation which are absent in normal cell-free DNA. That is very important to remember. And DNA is preferred over serum because blood lysis during the serum preparation could release plasma non-neoplastic cell. Therefore, we use the plasma DNA than the serum because there's a clotting process involved which can get a few normal DNA. It is unstable. It is collected using specialized blood tube. Must be sensitive enough to pick up the low-level variants. Limited number of mutations detected. The methodology, they are single locus or multiplex assay using a real-time PCR or digital droplet PCR technique or an NGS-based technique. This is one of the examples of digital droplet PCR. In one machine, in 4 hours, you have the report. So this is a beautiful technique. Then clinical applications of liquid biopsy, mostly for screening, monitoring of disease, actionable target. When there is no hope with the tissue, you can use this technique. Like diagnostic, the recurrent adenocarcinoma in the lung, which is EGFR mutated, when the tissue diagnosis is not feasible, a liquid biopsy can detect the recurrence. Therapy, if over the period of time the patient develops resistance to EGFR TKIs, osimertinib can be used, which can deal the T790M mutation, which is a resistant mutation in EGFR gene in exon 20. Response to therapy, serial monitoring of level of mutation, and detection of clonal evolution. This is a FISH test for ALK EML fusion, because normally what happens, the fuse signal, 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 the f This is something important. If you see here, when you're using break apart, so if you see fused green and red, that means it's good. In a break apart probe, when you see there is a separation, it is broken apart or it is gone, they are separate out, that means they are there is a translocation event which is going on. So this one in the bottom is abnormal, whereas this is one in the top is normal. Then this is the fusion technique. We are seeing a lot of fusion probes. So in summary, the cancer patients, they have recurrent mutations which are characteristics. They can be chromosomal alteration, epigenetic events, or DNA mutation. Cytogenetics, which is the study of inheritance in relation to the structure and function of the chromosome, and gene rearrangements can be seen by PCR. FISH. PCR is used to amplify the target DNA, and sequencing will take you forward the target sequence, which can be used by various techniques and find out more and more molecular pairs. So the take-home message in my talk today and tonight, which is almost like 10:30, cancer is a heterogeneous genetic disease, and oncogenesis or carcinogenesis occurs over a period of time which is long enough than we thought in practicality in the most aggressive way by gradual accumulation of mutations. The driver mutations, they impact the cell with malignant potential, with acquiring more and more tumor mutational burden, and the hallmark of cancer acquired during the process of oncogenesis. So what we think is not that clear cut. It is going in a skewed way, and we are providing the reports for the oncologists and helping them in generating this high volume, high throughput molecular data and interpreting the data along with them for the benefit of the patient. We are the garden of paraffin and the RNA, DNA, and protein. This slide I inherited from my mentor almost 10 years ago. I always use with my high regards to him. And so acknowledge my talk to all my juniors, friends, and mentors throughout my academic year. Thank you very much for your